pick up an egg. If you don't have one in front of you, picture it in your hand right now. The weight of it, the cool shell, the faint rattle of the yolk inside. That small object contains about 186 milligrams of cholesterol. For 40 years, a government pamphlet told you that number should terrify you. Here's the part almost nobody explains correctly. Your liver already makes roughly 3 to 4 times more cholesterol every single day than that egg could ever add. And when you eat more, for most people, your liver simply makes less. This is not a wellness video. I'm not going to tell you eggs are a superfood, and I'm not going to tell you they're a heart attack in a shell. By the end of this, you'll understand exactly what your liver does. The moment that cholesterol hits your bloodstream, why the original fear made sense on paper but fell apart in real bodies, and why the honest answer depends on something about your own biology that most people have never been told to check. Let's start with where the fear came from, because it wasn't invented out of nowhere. In the 1950s, a physiologist named Ansel Keys, working out of the University of Minnesota, published research linking dietary fat to blood cholesterol and heart disease across different countries. His work became the scientific backbone of decades of public health advice. The reasoning looked airtight. Cholesterol clogs arteries, eggs contain cholesterol, so eating eggs should raise your risk. It's clean, it's logical, and it's the kind of reasoning that feels obviously true. It also turns out to be built on a shortcut that doesn't hold up once you look inside the actual machinery of the liver. And to be fair to Keys, his original 7-countries study, published in the 1970s, was more careful than the popular version of the story that followed it. But 2 other researchers at the time, Jacob Yerushalmi and Herman Hillebo, published a pointed critique showing that if you included all the countries with available data rather than the specific set Keys chose, the tidy relationship between fat intake and heart disease weakened considerably. That critique didn't get nearly the attention the original correlation did, and it's a useful reminder of something worth sitting with for a second. A correlation across countries, however striking on a graph, is not the same thing as watching what happens inside one person's bloodstream when they actually eat the food. Here's the shortcut. The body doesn't treat cholesterol in food and cholesterol in blood as the same pool of water simply flowing from one container into another. Your liver is not a passive drain. It's closer to a factory with its own production line, and that production line has a manager watching the loading dock. Picture that factory floor. Cholesterol arrives at the liver from your intestines after a meal, packaged in tiny particles. The liver's job is to keep the total supply of circulating cholesterol inside a fairly narrow range, because cholesterol isn't a toxin sitting around waiting to do damage. It's a structural material. It's in every cell membrane in your body. It's the raw material for testosterone, estrogen, cortisol, and vitamin D. Your body needs it too badly to leave its levels to chance, so the liver has a sensing system. Inside liver cells, there's a protein called SREB2. Think of it as the floor manager standing at the loading dock, watching how much raw material is coming in. When dietary cholesterol goes up, SREB2 gets the signal and does 2 things. First, it tells the liver to slow down its own internal cholesterol production line, an enzyme called HUM-G-CoA reductase, the same enzyme that statin drugs target directly. Second, it adjusts how many LDL receptors sit on the surface of liver cells, which controls how quickly cholesterol gets pulled back out of the bloodstream and into storage. Eat more, and in most people, the factory throttles its own production and increases cleanup. Eat less, and the factory ramps production back up to keep the supply steady. This is why, in landmark feeding studies, including a well-known set of controlled trials from David Krzyzewski in the 1970s and, more recently, decades of controlled feeding work summarized by researchers like Maria Luz Fernandez at the University of Connecticut. Adding 1 or even 2 eggs a day to a healthy person's diet typically raises blood cholesterol only modestly, and often not at all. The liver is doing exactly what a well-run factory does when a supplier sends extra material. It adjusts output to match demand, so the loading dock doesn't overflow. Here's the part most people get wrong, though, and it's the detail that actually matters. Not every liver adjusts equally well. Roughly 1 in 4 people, researchers call them hyperresponders, have a floor manager that doesn't throttle production as efficiently. In these individuals, dietary cholesterol does raise blood cholesterol more noticeably. There's no blood test on a standard panel that tells you which group you're in. The only real way to know is to track your own cholesterol numbers before and after, meaningfully changing your egg intake over several weeks. That single fact, that this response is not universal, is the piece the original blanket warnings left out entirely, and it's also the piece that later loosened them. Which brings us to the official reversal, because this part is verifiable and it surprises almost everyone. In 2015, the U.S. Dietary Guidelines Advisory Committee removed the long-standing recommendation to limit dietary cholesterol to 300 milligrams a day. The committee's own language stated that cholesterol was no longer considered a nutrient of concern for overconsumption, specifically because the evidence connecting dietary cholesterol to blood cholesterol in the general population was weaker than earlier assumed. That's not a fringe opinion. That's the same body that used to be the source of the warning, updating its own position once the feedback loop mechanism in the liver was better understood. Quick, useful detail before we move on. Every bit of that 186 milligrams of cholesterol sits inside the yolk. The white is essentially none of it. It's almost pure protein, mostly a protein called albumin, roughly 6 grams of it, with no cholesterol at all. So if you've ever seen someone order an egg white omelet specifically to dodge cholesterol, now you know exactly what they're doing biochemically, and why it works if that's their specific goal, even though for most healthy people, it may not have been necessary in the first place. There's also a cooking detail worth knowing, because it's a separate chemical event from anything we've covered so far. When cholesterol is exposed to high heat for extended periods, deep frying, or scrambling at very high temperatures until the edges brown and crisp, some of it can oxidize into compounds called oxysterols. Research on oxidized cholesterol, including work summarized by nutrition scientists studying dietary oxysterols since the 1990s, suggests these oxidized forms may behave differently in the artery wall than intact cholesterol, potentially promoting inflammation more readily. The evidence here is less extensive than the feedback loop research we just covered, but the practical takeaway is simple. A soft-boiled or gently scrambled egg is undergoing a different chemical fate than one fried hard and crisp at high heat, independent of the cholesterol feedback question entirely. Now, before this turns into eggs are cleared, go eat a dozen a day. Slow down, because that oversimplifies it just as badly in the other direction, and there's a second, completely different mechanism that changes the picture depending on who you are. This second mechanism has nothing to do with your liver's production line. It happens further downstream, in your gut, and it was discovered by a cardiologist named Stanley Hazen at the Cleveland Clinic. Egg yolks are one of the richest dietary sources of a compound called choline. Choline itself is essential. Your brain needs it to make the neurotransmitter acetylcholine, and your liver needs it for normal fat transport. But certain species of gut bacteria can grab hold of choline before your body absorbs it and convert it into a gas called trimethylamine. That gas travels to the liver, where an enzyme oxidizes it into a compound called TMAO, trimethylamine oxide. Hazen's research, published starting around 2011 in the journal Nature Medicine and expanded in follow-up studies through the 2010s, found that higher blood levels of TMAO are associated with increased risk of atherosclerosis, the hardening and narrowing of arteries. Here's the rehook worth sitting with. This means the same egg can produce two almost entirely separate biological stories at the same time. One story runs through your liver's cholesterol feedback loop, which for most healthy people is remarkably forgiving. The other story runs through your gut bacteria's conversion of choline into TMAO, and that story depends heavily on which bacterial species happen to be living in your intestines. Something shaped by years of your overall diet, not by the egg you eat this morning. This is why two people can eat the exact same omelet and walk away with completely different internal consequences. It was never really about the egg. It's about the combination of your liver's responsiveness and your gut's bacterial population, both of which are invisible to you and neither of which shows up on a nutrition label. So where does that leave the actual research on outcomes? Not mechanisms, but real people, tracked over years. This is where I want to be careful, because this is exactly the kind of claim that gets stretched in either direction by different camps online. A large meta-analysis led by Ying Rong, published in the BMJ in 2013, pooled data from multiple long-term studies and found no significant association between egg consumption and coronary heart disease or stroke in the general population. That's a real, honest finding, and it's a reasonably strong one because it draws on a large pooled sample. But the same body of research consistently finds a different pattern in one specific group: people with type 2 diabetes. In diabetic populations, several of the same long-term cohort studies, including data drawn from the Physician's Health Study, have found a more consistent association between higher egg consumption and increased cardiovascular risk. Researchers don't fully agree on why diabetes changes the picture, but the leading explanation ties back to exactly the mechanisms we just walked through. Insulin resistance appears to impair the liver's normal cholesterol feedback response, and diabetes is also associated with shifts in gut bacterial populations that can increase TMAO production. In other words, the two mechanisms we just built, piece by piece, are the leading candidates for explaining why the same food behaves differently in a different metabolic environment. Why would diabetes specifically change how the liver's loading dock behaves? The leading explanation involves insulin. Insulin normally helps regulate SREB2 activity alongside dietary cholesterol levels, and in insulin-resistant states, that regulation becomes less precise. The Flaw manager, so to speak, starts getting mixed signals about how much raw material is actually needed, and the throttling response we described earlier becomes less reliable. This is inference built on well-established insulin signaling research rather than a single definitive trial proving the exact chain in eggs specifically, and it's worth being upfront that this particular link is plausible and consistent with the data rather than fully nailed down. That's worth pausing on because it reframes the entire question. Are eggs good or bad was never a fair question on its own. The more accurate question is, how well is your liver's feedback loop working, and what does your gut do with choline? For most people without diabetes or existing cardiovascular disease, the honest research answer is that moderate egg consumption, commonly studied in the range of about 1 egg a day, does not show a clear, consistent link to heart disease in the largest pooled analyses. For people with diabetes, existing heart disease, or known high cholesterol responsiveness, the more cautious approach has more support behind it. There's another study worth naming specifically because it's one of the largest and longest of its kind. Frank Hu and colleagues at Harvard followed over 100,000 men and women for up to 14 years in the Nurses' Health Study and the Health Professionals Follow-Up Study, publishing their results in JAMA in 1999. Among people without diabetes, they found no meaningful association between egg consumption of up to 1 egg a day and coronary heart disease. Among people with diabetes, the association with increased risk showed up again. The same pattern we just discussed, now confirmed in a second, independent, large data set rather than a single study standing alone. Here's a detail that changes how you should even read a cholesterol number on a lab report, and it's rarely explained clearly. Total cholesterol and even standard LDL cholesterol are measuring the amount of cholesterol carried inside particles, but they're not directly counting the particles themselves. A researcher named Alan Snyderman, working out of McGill University, has argued for decades that the number of LDL particles, measured directly through a protein called ApoB, one copy of which sits on every LDL particle regardless of its size, predicts cardiovascular risk more precisely than the cholesterol content those particles happen to be carrying. This matters here because it connects directly back to the particle size shift we mentioned with egg consumption. If eggs nudge your cholesterol toward being carried in larger, more buoyant particles rather than creating more small, dense ones, the ApoB count may barely move even if a basic cholesterol number ticks up slightly. It's a more precise lens sitting quietly behind the number most people are told to worry about. Let's do the honesty pass now, because this is the part that builds trust instead of just excitement. What's strongly established? The liver's cholesterol feedback mechanism through SREB2 and LDL receptor regulation is textbook biochemistry, verified repeatedly across decades of metabolic research. What's established but modest? For most healthy people, dietary cholesterol from eggs produces a real but small shift in blood cholesterol, and importantly, some research, including work from Fernandez's lab, suggests that shift often favors larger, more buoyant LDL particles rather than the smaller, denser particles more strongly linked to arterial damage. That's a genuinely interesting nuance, but it's a modest effect, not a dramatic protective one, and it shouldn't be oversold. What's physically plausible, but still being actively researched? The exact size of TMAO's contribution to cardiovascular risk in otherwise healthy people, and whether it's a direct cause of artery damage or more of a marker that travels alongside other risk factors. Hazen's own research team continues to investigate that causal question directly, and it isn't fully settled. Now here's something that almost never gets mentioned in either the fear-based version of this topic or the defensive version, and it's a completely different consequence worth adding, not a repeat of what we've already covered. Eggs are also one of the very few natural food sources of lutein and zeaxanthin, two pigments that accumulate specifically in the retina of your eye, in a region called the macula. Research from the age-related eye disease studies, large trials funded by the National Eye Institute, has linked higher intake of these two pigments to a reduced risk of age-related macular degeneration, a condition that becomes significantly more common after age 55, which is exactly the audience thinking about this question most seriously right now. What makes egg-derived lutein and zeaxanthin worth mentioning specifically, rather than just pointing you toward leafy greens, is that the fat naturally present in the yolk appears to help your intestine absorb these pigments more efficiently than when they arrive from a low-fat vegetable source alone. A finding reported in small feeding trials comparing spinach-only intake against spinach eaten alongside eggs. This isn't about cholesterol at all. It's a completely separate nutritional contribution sitting inside the same food, and it's one of the reasons the blanket "eggs are dangerous" framing missed something real on the other side of the ledger. One more honest note on the TMAO pathway, because leaving it out would make the picture unfairly one-sided against eggs specifically. Hazen's own research group has found that red meat, through a compound called carnitine, and certain kinds of fish, through a compound that converts to trimethylamine even more directly, can produce comparable or in some cases larger TMAO responses than eggs do. Choline from eggs is one contributor among several dietary sources feeding the same gut pathway, not a uniquely dangerous one. That distinction matters, because it's easy to walk away from this thinking eggs carry some special risk that other common foods don't also carry through the same bacterial conversion. There's a completely separate reason eggs matter specifically for an audience past 55, and it has nothing to do with cholesterol or TMAO at all. Starting in your 5th and 6th decades, skeletal muscle naturally begins to decline in a process researchers call sarcopenia, and one of the countermeasures repeatedly supported in aging research, including work published by Douglas Padden-Jones and colleagues studying protein synthesis in older adults, is getting enough high-quality protein containing sufficient leucine, an amino acid that directly triggers the muscle-building signal in your cells. Eggs are what nutrition scientists call a complete protein, containing all 9 essential amino acids in proportions the body uses efficiently, and the yolk specifically also supplies vitamin D and B12, two nutrients that become harder to maintain at adequate levels with age, particularly B12, since stomach acid production naturally declines over time and reduced acid impairs B12 absorption from food. None of this cancels out anything we discussed about cholesterol. It's a separate ledger entry sitting inside the same food, and it's arguably the most directly relevant piece of this whole topic for someone specifically in your age range. One more piece of real variation, and this one has nothing to do with your body at all. It's about the egg itself before it ever reaches your kitchen. Not all eggs carry an identical nutritional profile. Research on hen feeding, including studies out of the University of Guelph in Canada, has shown that hens fed diets enriched with flaxseed or fish meal produce eggs with meaningfully higher omega-3 fatty acid content, sometimes several times higher than a conventionally fed hen's egg. That's where the omega-3 enriched label on some cartons actually comes from. It isn't marketing fluff. It reflects a real, measurable shift in the fat composition of the yolk based on what the hen ate. It doesn't change the cholesterol or choline story we just spent most of this explaining, but it's a genuine, separate variable sitting on the shelf next to the eggs you'd otherwise treat as identical. Let's bring this back to your factory floor for a moment, because there's one more layer worth adding to it. The factory doesn't just respond to how much material shows up. It also responds to what else is happening in the building at the same time. If you're eating that egg alongside a plate of processed meat and refined starch, your liver and your gut bacteria are dealing with an entirely different chemical environment than if that same egg shows up next to vegetables and olive oil. This is part of why large nutrition studies increasingly measure whole dietary patterns instead of isolating single foods. The Mediterranean-style pattern, studied in the PREDIME trial out of Spain, published in the New England Journal of Medicine in 2013, is a good example. Eggs were never isolated as the villain or the hero in that trial. The overall pattern of the diet mattered more than any single ingredient sitting inside it. So what do you actually do with all of this? Let's move from mechanism into action, because understanding your own factory floor should change what you do tomorrow morning, not just what you know. First, if you have no diagnosis of diabetes, no existing heart disease, and no known history of your cholesterol spiking dramatically with dietary changes, the current evidence supports treating whole eggs as a reasonably neutral, moderate food rather than something to fear. Commonly studied at around 1 egg a day, sometimes more depending on the rest of your diet. This connects directly to the feedback loop we walked through. For most people, that loading dock adjusts, and the factory keeps output roughly stable. Second, and this is the one almost nobody tells you, if you genuinely want to know whether you personally are a hyperresponder, get a baseline lipid panel, then eat consistently for 4 to 6 weeks with a clearly higher or lower egg intake than your normal pattern, then retest under similar conditions. This isn't guesswork dressed up as science. It's the same logic researchers use in controlled feeding studies, just run on a sample size of 1. It will not tell you your absolute risk. It will tell you which category your own liver falls into, which is more useful to you personally than any population average. Third, pay attention to what surrounds the egg on your plate, because of that gut bacteria mechanism we covered. Diets consistently higher in fiber and plant diversity are associated with different, generally less TMAO-favorable gut bacterial populations in the research coming out of Hazen's group and others studying the gut microbiome. This doesn't mean a fiber-rich breakfast cancels out TMAO production. It means the bacterial environment processing that choline is something you have some influence over across months and years, not something fixed the moment you crack the shell. Some of the specific bacterial genera repeatedly flagged in this research, including certain species within the Prevotella group, appear more efficient at the choline to trimethylamine conversion, while gut populations shaped by long-term plant-heavy eating patterns tend to favor different, less TMAO-producing bacterial communities. Nobody is going to test your stool for this before breakfast, and you don't need to. The practical version of the finding is simply that the choline in an egg doesn't have a fixed fate. It depends on tenants you've been feeding for years before that egg ever arrived. Fourth, if you do have type 2 diabetes, established heart disease, or a documented hyperresponder pattern from your own testing, the more cautious pattern, commonly discussed in the research as a lower frequency, alongside closer attention to overall dietary pattern, has clearer support specifically for your situation. This isn't a moral judgment about eggs. It's matching the food to the specific metabolic environment it's entering, exactly the way the diabetes research we discussed suggests you should. Some clinical guidance discussed in diabetes nutrition research has centered on roughly 3 to 4 eggs a week rather than daily intake as a reasonable middle ground for that specific population. Not as a rigid ceiling, but as a starting point to discuss with a physician who can see your actual lipid panel and full medical history, which matters far more than any general number a video can hand. Fifth, if you're already taking a statin, it's worth knowing that statins work directly on that same HUMG-CoA reductase enzyme we discussed at the loading dock. They suppress the liver's own production line by design, the same lever your body pulls naturally when dietary cholesterol rises. Some research on statin users has found that dietary cholesterol matters even less for their blood levels than it does for the general population, because that production line is already being held down pharmacologically. That's a conversation worth having with your own doctor, rather than a rule to apply on your own. But it's the kind of detail that explains why the same nutrition advice doesn't always fit every reader equally. And let me be honest about what none of this will do. None of this will turn eggs into a treatment for existing heart disease. None of this means the lutein and zeaxanthin in eggs will meaningfully reverse macular degeneration that's already progressed. The research supports risk reduction over years, not repair. And knowing your hyperresponder status from a home experiment isn't a substitute for an actual conversation with your physician about your full cardiovascular risk picture, especially past 55, when that picture includes far more than just what's on your breakfast plate. Before we close, quickly. If you've made it this far, you clearly care about getting the real mechanism instead of the headline version. So tell me in the comments which side you came in believing, and whether the liver feedback loop or the TMAO gut pathway surprised you more. I read these, and that one detail helps me know what to explain more carefully next time. So go back to that egg in your hand. It looks exactly the same as it did at the start of this. But you're not looking at the same object anymore. You're looking at a small package of raw material arriving at the loading dock of an organ that's been quietly adjusting its own production for your entire life, long before anyone told you to worry about it. You're looking at a handful of choline molecules that may or may not become a gas, depending on which bacteria happen to be living in you right now. You're looking at two pigments heading toward the back of your eye, doing work that has nothing to do with your arteries at all. The egg didn't change. What changed is that you can now see the machinery running underneath the question, instead of just inheriting someone else's 50-year-old answer to it. Think about how strange that really is. A single food sat at the center of public health advice for half a century, repeated by well-meaning doctors, printed on pamphlets, absorbed by entire generations as settled fact. And the honest answer was never a flat yes or no. It was a set of feedback loops, a population of bacteria, an enzyme with a name most people will never need to say out loud, all quietly deciding the outcome differently for every single person holding that same shell. That's not a failure of nutrition science. It's just what happens when something that looks simple from the outside turns out to be built from parts, and understanding any one of us properly means being willing to open it up rather than settle for the label on the box. So the next time someone tells you with total certainty that a food is simply good or simply bad, you now have a fair question to hand back to them. Good or bad for which mechanism, in which body, processed by which bacteria? That question, more than any single answer about eggs, is the thing actually worth carrying out of this video.